In this class, we'll discuss about HDL metabolism. This HDL molecule will help in reverse cholesterol transport. In this reverse cholesterol transport, this cholesterol will be transported from peripheral tissues to liver, and in liver this cholesterol will be metabolized and forms bile acids and bile salts, and these are excreted through bile. And as this HDL molecule is taking up cholesterol from peripheral tissues, so it will prevent deposition of cholesterol in the peripheral tissues. So, and it will prevent atherosclerosis also. That's why this is called as a good cholesterol. Now, before going into proper HDL metabolism, we should learn about few terms. One is nascent HDL. This nascent HDL will be secreted from liver and intestine, and these HDL molecules will have ApoC and ApoE also. The liver HDL will have ApoC and ApoE, but intestine HDL will not have ApoC and ApoE. They will acquire ApoC and ApoE from liver HDL. And this nascent HDL will be secreted from liver and intestine, and they will have phospholipid, cholesterol, and ApoA1 molecule, and this will be in discoid shape. Next one is we should learn about LCAT. This LCAT is called as lecithin cholesterol acyl transferase. This lecithin cholesterol acyl transferase enzyme will help in transfer of fatty acid from C2 of lecithin to cholesterol. Due to this transfer of fatty acid from lecithin to cholesterol, they will form two compounds, that is cholesterol ester, and another one is lysolecithin. And this cholesterol ester which is formed will be going into core of nascent HDL, and due to entry of this cholesterol ester into the core, and they will form hydrophobic core, and due to this entry of cholesterol ester into the core, they will form HDL3 molecule. Nascent HDL is converted into HDL3. What happens to this lysolecithin? This will be attached to plasma albumin, and it will enter into the circulation. Lysolecithin will be entering into the circulation. Next, we will discuss about SRB1. What is SRB1? They are called as class B scavenger receptor B1. It has dual role. That means based upon the presence of this SRB1 in the different tissues, it has a different role. Whenever this SRB1 is present in liver and steroidogenic cells, they will have different function. What they will do, they will bind with HDL molecules via ApoA1, and this binding of HDL with the through ApoA1 onto the liver, SRB1 receptors causes delivery of cholesterol ester to liver cells and causes metabolism of cholesterol. Next, whenever this SRB1 is present in peripheral tissues, they will cause cholesterol efflux, from cells to HDL molecule, and this HDL molecule will transport cholesterol to liver for metabolism into bile acids and bile salts, and through bile they will be excreted. Now we'll discuss about HDL2. We have seen that nascent HDL through the action of LCAT, cholesterol esters are formed, and this cholesterol esters will enter into the core and they will form HDL3. HDL3. And these HDL3 molecules further accepts cholesterol from tissues, and they will increase in size of HDL molecule, and they will become less dense, and these less dense molecules are called as HDL2 molecules. That means these HDL molecules are greater in size than HDL3 molecules. And these HDL molecules will reach liver, and in liver they undergo hydrolysis. They will undergo hydrolysis through hepatic lipase. And in endothelium we have a lipase, and this endothelial lipase will also cause hydrolysis. And now after hydrolysis, this HDL2 will be again converted back into HDL3. This interconversion of HDL2 to HDL3 is called as HDL cycle. Now we should learn one more term. Sorry, this is 4, and one more term, that is pre-beta HDL. Now, what is this pre-beta HDL? Whenever HDL2 molecule is reaching liver and it is donating all the cholesterol, triacyl glycerols to the liver, then this HDL2 molecule will become a pre-beta HDL. How it will become a pre-beta HDL? In this free beta HDL, only free ApoA1 will be there. Only free ApoA1 is released, and this free ApoA1 has two fates. Through kidney they are excreted, and or this free ApoA1 will associate with minimum amount of phospholipid and cholesterol, and they will form a molecule called as pre-beta HDL. Pre-beta HDL. So whenever HDL2 reaches liver, this can be converted into HDL3, or this HDL2 can be converted into free ApoA1 by donating HDL2, donating all of its compounds, and it will can form free ApoA1. That free ApoA1 which is released will have two fates. In kidney, whenever it reaches the kidney, it is excreted, and or they will associate with minimum phospholipid and cholesterol, and they will form pre-beta HDL. And this pre-beta HDL is a most potent form of HDL, inducing cholesterol efflux from tissues. So this pre-beta HDL is a very important. Now we'll discuss about ATP bind. This is 5, this is 6. ATP binding cassette transporters. They are called as an ABC transporters. They will couple hydrolysis of ATP to the binding of substrate to transporter. And this ATP binding cassette proteins are of two types, ABC-A1 and ABC-G1. This ABC-A1 will transport cholesterol from cells to pre-beta HDL, and they will form HDL3 molecule. And this ABCG1, what they will do, they will transport cholesterol from cells to HDL3 molecule, and this HDL3 will become HDL2. So ABCA1 and ABCG1 has two different functions. ABCA1 will act on the pre-beta HDL molecules, and ABCG1 will act on HDL3 molecules. They will donate cholesterol to HDL3 molecules, and they will form HDL2. Next, we should learn about CETP. What is CETP? Cholesterol ester transport protein. Cholesterol ester transport protein. This CETP, what they will do, they will cause exchange of cholesterol and triacyl glycerols from other ApoB molecules. That means this is HDL2 molecule, this is HDL2 molecule, and this is either VLDL or LDL molecule. And this CETP will exchange cholesterol ester from HDL to VLDL molecule or LDL molecule, and they cause transport of triacyl glycerol from VLDL and LDL to HDL2 molecule. And this is done with the help of CETP. And after retaining this VLDL, after retaining cholesterol ester from HDL to VLDL and LDL molecule, they are transported to liver, and their metabolism they will donate cholesterol esters to liver, and these cholesterol molecules are metabolized. And this is how indirect reverse cholesterol transport is occurring. Reverse cholesterol transport is occurring with the help of VLDL and LDL molecule. VLDL and LDL molecule. These are the molecules we should learn about them before going into a proper cycle. Now we'll see the cycle. This is liver and this is intestine, and both will secrete nascent HDL. This nascent HDL will be in a discoid shape. This nascent HDL will contain phospholipid, cholesterol, and ApoA1 molecule. This is ApoA1. This is called as a nascent HDL. Nascent HDL. And this nascent HDL will be attached with the LCAT. This is LCAT. And this will be in discoid shape. Now with the action of LCAT, this cholesterol, free cholesterol which is present in the phospholipid bilayer will be transported to the core, and this transport of cholesterol ester, this LCAT will help in formation of cholesterol ester, and this cholesterol ester which is formed will be transported to the core of the nascent HDL, and they will form a molecule called as HDL3. This HDL3 will contain cholesterol ester and phospholipid, and they will have ApoA1 and LCAT. Now here is the peripheral tissue, and this peripheral tissue will have ABC A1 SRB1, and one more is ABC G1. Now this SRB1 will transport cholesterol from peripheral tissues to this HDL3 molecule, and ABC G1 will transport cholesterol from peripheral tissues to the HDL3 molecule. And this HDL3, after taking up of lot of cholesterol from the peripheral tissue, it is converted into a bigger molecule and lesser dense molecule with cholesterol ester and phospholipid. This is called as HDL2 molecule. Now this HDL2 molecule, with the help of CETP, this is CETP. With the help of CETP, they will transfer cholesterol esters from HDL2 to VLDL or LDL molecule, and they will take up triacyl glycerols from VLDL or LDL molecule to the HDL2 molecule. Now these HDL2 molecules will go to liver. And in liver also we have SRB1 receptors, and they will have ApoA1. This ApoA1 will bind with this SRB1 receptor, and cholesterol ester, and phospholipids will enter into the liver, and they are processed into bile and bile acids and bile salts, and they are excreted through the bile. Now, 50% of process will occur through this mechanism. Another 50% will convert with the help of hepatic lipase. This hepatic lipase will metabolize this HDL2 molecule, because they will have triacyl glycerols also. These HDL2 molecules will have triacyl glycerols, and this hepatic lipase will convert these triacyl glycerols, and this HDL2 molecule is converted into HDL3 molecule. This hepatic lipase and endothelial lipase will help in conversion of HDL2 molecule into HDL3 molecule. And during this process, this HDL2 molecule will donate cholesterol ester, and phospholipids and triacyl glycerols to the liver, and they will release only ApoA1. This ApoA1 is poor in lipid, and this ApoA1 will grab only minimum amount of phospholipid, and cholesterol will be attached to this ApoA1. This is called as pre-beta HDL. This is pre-beta HDL. And this pre-beta HDL will take up cholesterol from the ABCA1 receptor, and they will be converted into disc-shaped nascent HDL molecules, and again this nascent HDL molecule will further go into the cycle. This is how HDL metabolism will occur, and this HDL molecule will take up cholesterol from macrophages, that is foam cells, through diffusion, or they will use ABC transporters, and they will prevent atherosclerosis. And this HDL molecule will have all the apolipoproteins, and this HDL molecules, they will donate apolipoproteins to chylomicrons and VLDL molecules. And this HDL molecule will be secreted from or synthesized from liver and intestine. The liver HDL will have ApoC and ApoE. Intestinal HDL will not have ApoC and ApoE. They will acquire from liver HDL. So this is about HDL metabolism, which help in preventing deposition of cholesterol in the peripheral tissues and prevent atherosclerosis in a person. Thank you.